if we were live and in person. So we're live and via video. So Kaleb will be speaking to us today about the GLORIA project, the Great Basin group, and GLORIA is the Global Observation Research Initiative in Alpine Environment that was started by a group in Austria. Kaleb is the field coordinator and board member for the GLORIA Great Basin group. He's originally from San Diego and did his undergraduate work at UC Santa Cruz. He's currently a third-year graduate student in the Department of Plant and Microbial Biology at North Carolina State University in the Sheth Evolutionary Ecology Lab, and where he's studying alpines and their responses to climate change, etc. So without further ado, take it away, Kaleb, and we're all looking forward to seeing and hearing about your research, and you're taking us visually to these wonderful alpine environments.
Okay, fantastic. Yeah, let's go. Let me just get my screen set up here. Just need to do this, and you're gonna see this just for a second. And then this took us a little bit to work out earlier. I need to switch. I know you can see the presenter view, and I just need to do this. And now I think you are seeing just the title slide. Everyone, thumbs up?
Yep.
Okay.
And also, if people have questions, please put them into the chat. We'll be monitoring the chat. So, so again, just put your questions there and we'll get to them. Thank you.
Totally. Okay. Yeah, thanks so much for having me today. Once again, my name is Kaleb Goff. I'm here in Raleigh, North Carolina today, but I think as Bart said really well, we're going to go on a little adventure together. So the title of my talk is Sierra Nevada and Great Basin Alpine Plant Communities in a Changing Climate. The title slide here is from this summer's GLORIA Great Basin field forays in the Sweetwater Mountains. So you can see let me just see if I can get a pointer here. You can see I think you can see this pointer. You can see Mono Lake is right here. Here's kind of like the Yosemite area. I think that's like Mount Dana right there. Here's the Sawtooth Range. So if you're familiar with this area. And yeah, I just want to give a huge shout out to the Eulers, to Michael and Ellen for kind of getting me in here. And I'm just grateful to drop into your all's community in this way. We're all these kindred alpine and just plant loving spirits. So yeah, the goal of this talk is just to entertain you first and foremost, and to inspire you and sort of drum up some of that wonder. And then to introduce you to some of my work and some of the work of our group in GLORIA Great Basin and of our lab group here. So I want to start with this, which is a quote that I try this time of year really to live by, written by this wonderful writer, David Gilligan. He says, how many, sorry, my bar is in the way here. He says, how many mountains does it take to reach the high country of the mind? We often glimpse it, but how do we get there? What does it take to bring the clarity of our experience of the mountains into all aspects of our lives? So this is something that, especially this time of year when it's winter and I'm in my office and I'm dreaming about the high country and dreaming about summer, that I'm sort of thinking about. And then David Gilligan writes, it is early evening, midsummer in the mountains. The sun disappears behind the high granite peaks. The air cools. The snow that remains here refreezes. Wildflowers, which stood boldly open in the bright day, Kalmia, Dodecathion, Potentia, shrink back as the light grows pale and the air cold. The wind that has whooshed and swirled through this place all day long lies down to rest. The land is hushed. I pull on long pants, 2 wool shirts, a warm hat, lace up my beat-up shoes, and set out up wide granite slabs for the alpine world. So just a little start, and here we go, lacing up our shoes, even here on Zoom today, and heading out together into the alpine world. And if you haven't checked out this fantastic essay by David Gilligan called The High Country of the Mind, check that out. So just a little bit of introduction about me. I kind of got my start as a botanist as a 18-year-old in Santa Cruz, traipsing around in the redwood forest and in the meadows, and going out into the Bay Area and going botanizing, going up to the Hamilton Range and seeing these two plants. Ended up working in Kathleen Kay's lab at UC Santa Cruz, sort of studying these two plants. They're each other's closest relative. So there's all these interesting questions about plant speciation, how we get more beautiful things. And it took me quite a while. This is sort of just came out. So if you're interested in kind of seeing more about this work, it's there. But it took a long time for me to get to this kind of becoming a researcher, partly because I got sidetracked into this other venture. And that other venture was really thinking about how to do a lot of good in the world, and meet a lot of people where they were. And one answer for me to that question was to do these free walks and talks and programs as a ranger naturalist in Yosemite National Park. And I spent a lot of time there sort of trying to inspire people and meeting people where they are and seeing how people's stories interacted with that place. Led a lot of botany walks and bird walks and things like that, and sort of tried to walk this high ridge between art and scientists, the kind of naturalist way. I spent a lot of time walking around in alpine places like this and taking people there. This is the summit of Gibbs, if you're familiar with the Yosemite area. And here's Mount Dana, which is a popular place to look at alpine plants in Yosemite National Park. Walking around in these places really made those questions about the alpine get a lot louder in my mind. So like, how are alpine plants doing with climate change? Why is this plant here and not there? Why are there so many beautiful, interesting alpine plants to see? All those things. And so I would say, part of the kind of organizing factor behind my work is understanding why there are so many beautiful things, understanding the origination and maintenance of biodiversity, thinking about biodiversity changes with climate change, being in these places and sort of bearing witness to them. I'm especially interested in mountains and in these heterogeneous places with these compressed climate gradients across short distances, so places where there's a lot of elevation to traipse over. And this is kind of a way that I've kind of tied together my passion outside as a naturalist with some of my academic work and professional work. So this is where we're going today to talk about some of the alpine biodiversity that you can see in plants. To say nothing about sort of all the other cool stuff that's up there, but that's for another time. So let's just walk around the world just quickly to some alpine places and just kind of start with a general definition of the alpine. I think of the alpine as being this place between the tree line and the highest elevations that are possible. So tree line is dictated by temperature. Tree line can be quite wavy. I think a lot of people have a good sense of that. These places where there are no more trees, where that are too high, have these really short growing seasons, have extreme temperatures, have low nutrient availability, high UV irradiance. And I want to just add that these places are super different from one another. So they all kind of share these common factors, but they're all really different, right? So like what you see in the Alps and in European alpine systems with these big meadows is quite different from these really rocky, flowery, garden-like places like we might see on the summit of Mount Dana, which is also quite different. All these places are alpine, but also quite different than what we might see in these kind of shrubby habitats at 4,400 meters in Tibet. We can go down to the southern hemisphere to think about the alpine. I'm just like super excited to show this slide because I just came back from a month in the alpine grasslands in South Africa. So here's the Drakensberg system right here. So these, plateau grasslands. There are subtropical and tropical alpine areas too, which aren't shown here. So this really, really heterogeneous group of places, kind of united by the fact that there are no trees there. And these kind of, there are all these cool growth forms that kind of predominate in those places. In the place we're going to be talking about, things kind of zooming into our neck of the woods, things look kind of like this around there. So this is the true Schwarzsmithian belly botany on the slopes of Mount Dana. And, when you're looking at these kind of places from far away, you're like, oh, it's a pile of rocks. Maybe there's some grasses. And then again, you kind of get into this zone and you start to really be blown away by just the diversity of these places and the diversity of forms. So yeah, how can someone not just be like absolutely enthralled by places that look like this? These places, of course, are dry and windy, and we think that they're threatened by climate change in some interesting ways. And one of the organizing sort of academic questions here, or bigger questions here, are whether or not alpine plant communities are robust to warming. So can the alpine plant communities that we know and love from the Sierra and Great Basin and elsewhere, can those persist into the future with this rapid and accelerating anthropogenic warming? One idea about that is this idea of an escalator to extinction, which is that as mountaintops, we're going to be really talking about just summits here. I've introduced the Alpine, but we're going to zoom in on the summits in a second. But that species on the summits, as it gets warmer, have sort of nowhere else to go. And that there's this, population extirpation and local extinction on summits because it gets warmer, and those species that are on the summits have sort of nowhere else to go. It's hard for them to get to the adjacent summit, and it's hard for them to sort of track their climatic niche, or the set of environmental characteristics that they need to survive in anywhere else because they're already at the top. The kind of rebuttal to this idea, or maybe the kind of the corollary to this idea, is that alpine plant communities might be sort of uniquely set up to deal with warming, and that's because they're really heterogeneous in terms of temperature. So there might be like, this is what we think of in terms of like these climatic belts, that there's kind of like this cake pattern. But actually, if you get up there and you really look at these thermal refugia on mountains, it looks more like this, right? Where there's like cold refugia and hot refugia, and alpine plants might just be able to move back and forth, left or right on a mountain slope instead of up and down. Even plants up here, right? Like if you're on this warm side, you might just need to go right over to there to get into a cool place, so that this microclimatic heterogeneity might buffer alpine plants from climate change. So two hypotheses, and we can start to think about those and evaluate those. But to do that, we need some data. So let's get that data. Bart really bravely and courageously jumped into this wild acronym for an international project called GLORIA. This acronym was made by people who I think probably speak many other languages besides English, but it's Global Observation Research Initiative in Alpine Environments. Started in Austria in 2004. And then brought over into North America by Connie Millar and others, or started in Austria in 2001 and brought over to North America in 2004. The basic idea is to monitor these alpine summits all over the world using the same protocol, using the same methodology, and then to be able to compare how alpine summit plants are changing over time as climate change continues to ramp up. Zooming into North America, and I'll also just say here, this looks like, wow, we've made a lot of progress. We haven't made a lot of progress in Africa, and this is kind of a thing that people talk about in science, right? Like where globe, you should maybe have big air quotes around global, because yeah, there's some really cool alpine areas here. Recently, we've learned a lot from Northern Europe. We've learned a lot from actually from the Snowy Mountains in Australia, and we have some other kind of GLORIA papers just starting to come out. So this is a work in progress. So zooming into North America, we're going to be talking about these target regions, and these target regions are sort of GLORIA speak for groups of peaks, and I'll show you the kind of what the groups of peaks look like. So here's North America. Here's some of the GLORIA regions that are either active or proposed. So we've got some kind of nice coverage here. GLORIA Great Basin is a nonprofit that manages these 8 target regions that are here in California and Nevada. So starting up north, just below Lake Tahoe in the Carson, kind of Friel Peak, Carson Range area, extending south to Death Valley, and then out encompassing some of these Great Basin ranges, and then out to Great Basin National Park in eastern Nevada. Those are the 8 target regions, and within these 8 target regions are 29 peaks that this nonprofit GLORIA Great Basin has been monitoring and working on since 2004. And this is my chance really to just give a huge shout out. I mean, there's people here who have worked on this for quite a long time. In 2004, I was in 5th grade. So just this kind of idea of how, these long-term projects can pass, but a huge shout out to all these people. They're all amazing, and I'm all just so grateful to be able to be part of this. And then also to say that this GLORIA Great Basin work happens because of hundreds of volunteer community scientists across 20 plus years. This is about to be the 20th year, 2004 to 2024. So many people out there. I know Michael and Ellen Euler are just fantastic contributors to this. And then, yeah, so huge shout out to this amazing collaborative work. Okay, so let's dive in a little bit. The first chapter of my dissertation is looking at the kind of biggest spatial scale and the biggest temporal scale and asking how the number of species on those summits has changed over time. I just wanted to show sort of the constancy of these places. So here's a place in the Dunderberg area, so kind of like Mono Lake area. Here's a plot from 2004 that we monitored, and here's a plot in 2014, 10 years later, right? So like there's the same plant is there and there. A lot of the same rocks are in exactly the same location. So there's a lot of constancy in these places. Just a little foreshadowing. So the place that we're going to be talking about, and I'm going to try to show you some results from, again, these 8 target regions. They are in this western part of North America. And just pulling out some of the kind of broad scale climatic data for these places, looking back to 1955. So that's the kind of historical period from 1955 to 2004. And then in gray, this shows the study period. So since we have all been working on these GLORIA Great Basin summits, 2004 to 2022, sort of showing some of the warming trends. So these places are definitely getting warmer, showing the variability of precipitation in this region. If you're a Californian, you sure do know a lot about that, right? You have even just the period I worked in Yosemite, we had the drought from 2011 to 2015, this, millennia drought. And then 2017, we had like 215% of average. And then we had some more kind of average years. And then last year was wild. So really this kind of spiky thing. And then climatic moisture deficit is a way to just get at this interaction between temperature and water. And again, sort of hard to draw some real strong patterns out of there. But just thinking about, so what are the kind of climatic conditions in these places? These are definitely arid ranges relative to other alpine areas globally. I just want to introduce you to some of the target regions, because the target regions are just like, they're all stars. They're just beautiful. So here we're in the White Mountains. So we're looking down into the Owens Valley. Bishop, California is would be down here. This is the White Mountains, and these are the silicaceous substrates. So these are like metamorphosed volcanic rocks. Really interesting. Here's some really sad GLORIA field biologists out there. Here's just one of the, I just want to pull out some of the emblematic taxa. Caloptridium umbulatum, Portulacacea, and talk about some families, but amazing plant that's up there, just incredible. So this is one of the target regions in the White Mountains. Another target region in the White Mountains is this one, the Dolostones, which are these metamorphosed limestones. In the back of the photo here, you can see the Great Basin bristlecone pines and limber pines, so some of the oldest non-clonal living things on Earth. Very different, right? Same mountain range, and you can see where some of those silicaceous rocks are mixing in up here, but these two very different places inside of one incredible mountain range. I won't say too much about it, but the White Mountains are just such a special place. Here's one of the kind of emblematic taxa there, Townsendia condensata, Asteraceae, something that just kind of makes you fall to your knees when you see it in summertime there. Zipping over to the other side of the caldera, into the Sierra Nevada. Here we are in the Mount Langley target region, which has some really high peaks. Here's some very intrepid GLORIA community scientists and GLORIA tiers, we sometimes call them. And here's a plot where it just has rocks. So that happens when you're working in the Alpine. And again, something that just is striking to me, and I love to notice and point out is, right, this just looks like, man, there's no plants here. And then right at the base of this rock where we're working is one of our, again, emblematic Alpine taxa, the sky pilot, Polemonium eximium, Polemoniaceae. So these are these unglaciated plateaus going to another target region in the GLORIA scope. Up there's Lake Tahoe in the corner of the frame there. Here's these decomposed granitic soils with a whole bunch of interesting plants. And here's the Mount Rose buckwheat, something that we see a lot. And these are these Krumholtz mats of whitebark pine. So another GLORIA target region. And then another GLORIA target region that we visited just this last year, and this comes from the title slide, again, Sweetwater Mountains, which are just to the north of the Bridgeport Valley. And then, oh man, this plant that just blows my mind every time I see it. a Crepus nana. So just want to show you 5 of the 8 target regions, and hopefully you can see just through the photos and some of the plants that are there, some of the similarities, but also to say even amongst these 8 target regions in this sort of relatively defined geographic area, there's a lot of heterogeneity just in these sites. So what I want to show you is some of the results from the first chapter of my dissertation, which is thinking about how the number of species on these summits has changed over time. So to do that, I want to just talk about the methodology for a moment. So we have these 8 target regions. In each target region, there are 3 or 4 peaks, and this is dictated by the International GLORIA Protocol. So there's 3 to 4 peaks within each target region. They're arranged across this elevation gradient. The peaks within the target regions are. So there'll be like a peak that's close to the tree line, and a peak that's kind of intermediate, or maybe 2 peaks that are intermediate, and then a peak that sort of represents the highest area in a mountain range. These peaks are chosen really specifically to avoid recreation traffic. We try to go to places that are not like on every peak baggers registry ever, because we're trying not to have people walk there. And then so we have 29 peaks across these 8 regions. Each of these peaks have 4 aspects, north, south, east, and west, and then 2 contours, 5 meters and 10 meters of vertical distance from the summit. So we actually measure down and we run out these strings. And that gives us these 8 plots. So 1, 2, 3, 4, 5, 6, 7, 8 plots. And that's sort of our big, and then in each of these plots, we count the number of species on each time point in each peak. I did something a little bit different. I kind of collapsed this a little bit more with the analysis, but I just want to show you the overall structure of this data. Before I jump straight to the results, I want to say once more, just to refresh, that these changes in Alpine Summit plant communities we don't know what's happening with them right now, and there's reasons to think that they might lag, so they might be slower or faster, some of the processes there. So like new things showing up might be lagged or might be expedited by certain factors. The establishment of new plants in the Alpine might lag, and then this extinction of plants that are already there might lag, or the pointed arrows show the lags increasing. So microrefugia might make extinctions take longer to happen. I want to introduce you just to a few properties of these ecological communities. So richness, we're going to be talking about. It's a really basic measure of biodiversity that just says what are the number of species that are there. Turnover, I want to talk just briefly about, which is sort of thinking about species gain and loss and the difference between two species lists. So if you like go to a summit in 2004 and then you come back 5 years later in 2009, how different are those 2 species lists? How much turnover has there been? And then abundance, I'm not going to talk too much about abundance, but just to say, it is important to know the number of individuals. And if you've kind of been working in this space, or you think a lot about this kind of idea, you think about, combining richness and abundance into some of these biodiversity metrics. And we're not going to get into that today, but just to say that those exist. So we could have richness increases, right? So maybe things are getting warmer in the Alpine, and that's making it easier to live in the Alpine. And so more things are living in the Alpine than ever before because it's warmer there, and it's easier to be a plant in those harsh environments. Maybe we could see richness decreases. We could see richness decreases, perhaps, again, just to use the same example from before, we might see richness decreases if it's getting harder to be a plant in the Alpine, or if the establishment lags are long and the extinction lags are short, right? So if there's population extirpations and population level extinctions on the summits, but things from lower elevations are not getting there as quickly, we might have a species richness decline on those summits. And then there's also, this potential that we don't see a change, and that might be because the climate hasn't changed enough. That might be because we haven't measured those places long enough, or it might be due to some kind of resistance or resilience in these communities. So without further ado, I know you're dying to see the next slide, which is showing the number of species. So the y-axis in these 8 plots, each one of these squares is a target region, and the number of species is shown on the y-axis. On the x-axis are the years. These peaks get surveyed once every 5 years. And then the triangles are the summits. And again, those summits within each target region are arranged by elevation. And so I just ranked those. So 1 would be the lowest site, so closest to the tree line, and then 4 would be the highest site, so closest to the upper elevation. And one thing you'll notice from here, if we just look, let's say over here in the upper right, in at the Mount Langley target region, there are these 4 summits, and the site that's way up on the shoulder of Mount Langley itself, the highest summit, has very few species, right? And then those lower summits have a lot more species. So this is what we expect. We expect that species richness decreases with increasing elevation. But the point of this figure is to say that when we looked at all of these, the number of species, and we tracked them over time across all these target regions, we didn't find any statistically significant changes in species richness over time. So according to this statistical model, and I'll say that you do see some kind of modest increases in species number, but across all these, we conclude that species richness hasn't changed over time, or it hasn't changed enough to exceed the amount of variability in species number from year to year. So kind of surprising. So just want to hold that for just a moment. That's one of the kind of results from GLORIA Great Basin, kind of looking at the biggest spatial scale and the biggest temporal scale. So all the years, all the peaks. I want to just add methodologically that each one of these triangles just shows, collapses the contours and aspects. So I just use the number of unique species on the peak. So yeah, so this doesn't include aspect changes. Are the aspects different, and are the 2 contours different? I did look at those, and I can speak to those if people are super interested in that. So the second question we might ask from this is whether we're seeing no change in richness because we actually find exactly the same things year after year, right? I showed that picture of like the same rock in the same place, or like that same Ericameria shrub in the same exact place in the same plot, a decade apart. Is it that we actually find the same thing? Or are we seeing no change in species richness because there's some gains, some species are being added and some species are being lost, and those are balancing each other out? To get at that, I took sequential lists. So I took 2 species lists that were on the same peak that were separated by 1 survey, so by about 5 years, and compared those, and I calculated the proportion of species that were different. So what this y-axis turnover shows is, like, for this point, for example, right here, this is 1 peak within the Carson Tahoe target region, and this peak between two surveys had about 21% turnover. So the difference between those 2 lists was about 21%. Across all of these target regions and across all these peaks, there was
So, sorry,
12.7%
sorry.
turnover. Like some
Go
people
on.
can try and mute if you don't mind. Thanks. So just want to reiterate, this shows the turnover, so the difference between sequential surveys on the same peak for each target region. So the big colored dots show the average for each target region, and then the small dots show the difference between 2 sequential surveys. And the average across all of this is about 12.7%. So on average, across all the GLORIA Summit Area section surveys, across all the peaks, we have about 13% difference between 2 surveys, between any 2 given surveys. My question for you is that a lot or is that a little? I can talk about kind of some of the ways that we might get at whether that's a lot or a little, but just like, how does that strike you? Does it seem like, wow, 13%, like that's a big difference, or 13%, I'm not sure. And again, these are peaks. Some of these peaks have, 70 species, and some have 8 species. So the reason to use this proportional turnover is it allows us to compare peaks that have different numbers of species. Okay, so just a little bit of kind of wrapping up there. There's a bunch of future questions here, so we might try to ask about these gains and losses. Oh, I should say the kind of bow to put on this is to say that there's no change in species richness because of a balance of gain and loss. So 13% turnover to me says that there's differences between lists. Some species are being lost, some species are being gained, and that balance of gain and losses and that turnover is sort of hidden behind no change in species richness. So that's kind of the answer to that question. And I'm happy to, I have a pocket slide that someone can hopefully ask me about all the kind of taxonomic cleaning and making sure that we know that we're actually have these clean species lists. Because if you've done this kind of work, you know that that is super difficult. So next questions we might want to ask of this data, or that I would love to ask about this data, is sort of thinking about these, grouping these species that have showed lots of gains or species that have showed lots of losses in terms of their shared traits. So we might think about just functional groups, forbs, trees, shrubs, graminoids, and cushions. We've done a little bit of that and thought a little bit about the abundance change and the turnover change, which I'm not going to show here, but will be in the paper, hopefully later this year. And then another thing that I've done, but I'm not going to show here, but I think is really interesting and could be kind of included in future work, is thinking about whether certain species are clear winners and losers. And of course, that takes this sort of sharp botanical eye. And it requires, I think, looking at a finer data scale. So I've just shown you the biggest data scale, so those big plots. We also have these quadrats where we look at quantitative abundance, and we look a little bit closer. So lots more here, and I want to leave this chapter there because for me, this got me quite a bit more interested in traits. Right, one way we can think about biodiversity is that it has multiple facets. There's multiple ways to think about biodiversity. We might think about taxonomic diversity, right, which is what we just talked about. What species are there? What's the number of species that are there, using these sort of biological species concepts. We might think about what phenotypes or what characteristics, what traits are in a community. So are there lots of tall plants? Are there lots of short plants? Are there lots of plants with thick leaves? Are there lots of plants with thin leaves? So that's, the traits are a facet of biodiversity. There's this functional facet. And then we might also think about the evolutionary relationships of the plants there. So the phylogenetic diversity. And so this first chapter is all about taxonomic diversity. That's what I've just shown you. And then I want to transition to some questions that have been really sneaking up on me and getting into my mind, that are all about functional trait diversity and the differences in what traits are there. So that's where we're going to go now. So if you're going to group species based on their traits, why not just go out into the field and actually measure the traits that these plants have? So here's just, a way to think about what are some of the functional traits that are out there. We have plants that have short leaves, but tall inflorescences, right? So here's a plant that is keeping its leaves close to the ground, but putting its flowers and fruits up into the sky. That's kind of interesting. We have lots of plants in the Alpine with super hairy, weird, thick, thin, wild leaves. So all these different leaf morphologies. We might have plants with different seed masses. That's not a trait that I measured here, but just to sort of think about what are some of the traits. We have plants like these that really also have a different way of being. They have really thin leaves. They have clasping leaves. They might have certain nutrient qualities in their leaves, if we get to that. So those are some of the functional traits that we'll talk about. And I think this kind of connects these facets of biodiversity. And so that's where I've been going with this work. So let me just back up and talk a little bit about why this might be interesting from a more kind of global perspective. Looking at these traits rather than looking at the species identities themselves helps us do some generalizing on a global scale, right? So we might say across the entire world or across all of North America, are a lot of the plants with thick leaves increasing in cover or increasing in their presence? Or we might think like our alpine plant community is getting taller on average, right? Maybe we expect that it's getting warmer and thus it's getting easier to be in the alpine. And so plants are willing to extend into the sky a little bit further. And so we can use functional traits that we measure to put plants on a spectrum from resource acquisitive, so resource conservative. So these are the plants that really are like holding it in and are preparing for a darker day, right? These are plants that they're short. They have small leaf area. Their leaf matter dry content is high. So they have these really thick leaves, small, thick leaves. Maybe they don't have a ton of nitrogen in their leaves. There are plants that are on that side of the spectrum. And we might also have plants that are on the more resource acquisitive side of the spectrum. So plants that are really like out there getting things done, gathering lots of resources, using them to do lots of things. They have these, another way to frame this is to say the resource conservative plants have a more slow kind of life history. And then the resource acquisitive plants are on this kind of fast side of the spectrum. And we can map these traits, so these traits that I just introduced you to, we can map them onto the plants that we see when we're walking around in the Alpine, right? So here's some of the resource conservative plants that you'll see out there in the Sierra Nevada and Great Basin when you're walking around. So like Eriogonum ovale, right? Look at those small, thick leaves and the short stature. Cymopteris cinerarius is almost hilariously resource conservative. It's a plant that like is doing its best rock impression. Raylardella, just a huge favorite of mine, but again, short stature, and then these kind of small, thick leaves. And then resource acquisitive plants, wow, they look really different. So here's some examples of resource acquisitive plants that you might see out in the Alpine walking around. So we can use these to ask how plant traits are shifting with global change. And we can use these as sort of important indicators in thinking about how these communities might be changing, might have changed, and might continue to change over time. And because we're not focusing directly on the species identities, although that is an important part of this, we can sort of scale these analyses up, and we can sort of think like what's happening in the Alpine in South Africa, what's happening in the Alpine in Norway, and what's happening in the Alpine in North America, and we can compare across those places because we can compare leaf thicknesses instead of comparing what species are there and what species aren't. GLORIA Great Basin, due to the very smart work of Connie Millar and Ann Dennis and Jim and Katie Bishop and many others, developed a cool protocol to think about what's happening between the GLORIA summit and the subalpine. You can't see the subalpine here. This is White Mountain Peak in Eastern California. So there's a GLORIA site here, and this, Jim Bishop made this figure. So this is like the area we're getting to know with the GLORIA, the summit area protocol, which I just showed. And we think that that species richness isn't changing much here. And I think one thing that might say to you is that the action is in this whole region right here, right? So the Alpine is more than just the summit. It's this whole, interesting, shifting, heterogeneous place between the tree line and those summits. So these downslope surveys are a way to get at what's happening in between the summit and the tree line. What they look like are these 100-meter long belt transects. So like at each one of these question marks, we would have one of these belt transects. So it's 100 meters long, it's 1 meter wide, and it has these 10 segments, and it's laid out on the landscape. So you can kind of imagine it tracking up. Right now in the White Mountains, we have 5 sets of these, and this is from a great paper, Smithers and Oldfather 2019, that gets at some of the spatial relationships inside of these downslope transects. But what if we could overlay some of our trait data on this and get at how are traits changing across this gradient in space, and how are traits changing across this gradient in time? That's sort of something that I'm working on currently right now. So to do that, we rely on our amazing, smart, super wonderful collaborators and friends, and we get out there and we survey these vegetation communities in this very systematic way. Then we have that community data set, and then we combine that community data set with some data about the traits. So we need to get out in the field and measure some plant traits. Yes, this is just as wonderfully simple as it sounds. We get out in the field with a ruler and we measure some heights. So we're in this case, I was interested in vegetative height. So that's the height of the tallest leaf or the highest vegetative material, and then generative height or reproductive height, which is again getting at this difference between how high plants are willing to put their inflorescences or infructescences into the sky. So we measured those heights. Then we clipped some leaves with scissors. Yep. Thanks to the INYO and others for helping us get the permits to do that in the right way. We take these leaves back to a lab and we scan them to measure the leaf area, and we weigh them to get the leaf wet mass. Then they get thrown in the back of my truck and they come here to the Sheath Lab in Raleigh. So they make a cross-country journey. And in the Sheath Lab, we dry them and measure their dry mass, and that lets us calculate specific leaf area. So that's the ratio of leaf area to leaf mass. And then it also lets us measure the leaf dry matter content, which is getting at sort of both of these are getting at that resource acquisitive to resource conservative spectrum. And so we wanted to do this for these 6 functional traits across all the species on the GLORIA Great Basin summits, and then we extended it from the summits to the downslopes. So we want to get trait data for all the species that are in that downslope area. This cannot be done by one person. It has to be done. So here's my amazing advisor, Seema Sheth. She's awesome. And here's Malia Reese, who was my field technician, amazing UC Davis undergrad, who was the field technician 2 years ago. And this year I had Sylvie Amstutz, who was a field technician from UC Santa Cruz. Here's us like pretending not to have a good time, even though we always have a good time. And here's us on top of the 3rd highest peak in California, White Mountain Peak. So yeah, we really love that area. It was a great time out there. So just want to show some of the plants that we collected trait data from. Here's Astragalus calicosus, and again, getting a sense of how we measure those things. So you can read the measurements off to me, but yeah, so we can see how high is the highest flower, and then how high is the highest leaf extending. So we record those. Here's this amazing Phacelia that grows way up there in the White Mountains. A plant that I'm just like generally obsessed about, Erigeron vagus, which is this really high elevation fleabane, high elevation erigerons just on some of the highest peaks there. Just blows my mind. If you've walked around the Alpine, maybe you've seen this plant, which is a Brassicaceae Anelsonia, also called dagger pod. So the silicles are just these like crazy cool black fruits. It's striking me literally in this moment that maybe that dark color helps to warm up the fruits so that they mature, or the ovules so they mature. I don't know. Someone should think more about that. Some other cool plants, some really interesting grasses. There's an endemic, or there's a really kind of interesting Polemonium species in the whites. Here's just another field photo, right? Here's us measuring Holsea algida at 4,000 meters on the shoulder of White Mountain Peak. This is a plant too that I think more people should be studying and thinking about. It's kind of violates some of the things that we might be expecting. We might be expecting that as you go up in elevation, plants get smaller and their leaves get thinner and more brittle, or maybe you expect that their leaves get thicker. But Holsea algida is just this like super tall, like fleshy, wild, like it's got these really sticky trichomes on it that smell like blue cheese. And yeah, it just kind of it's an interesting one to see this high up. You can see that there's not any other plants around here in this direct area that really look like that. Okay, so we collect all these traits, we take them back to the lab. This is at Crooked Creek, White Mountain Research Station. So huge, again, huge gratitude and shout out to them for putting up with us, and thanks to yeah, thanks to Tim and Dylan. Thanks to Dylan Neubauer for taking us around and like helping us figure out what plants we're looking at. And yeah, I just want to just really emphasize how many more people are behind all this. So yeah, huge gratitude to all those people. So here we are, we're scanning the leaves, we're weighing them on little scales that we brought, and we're getting the data entered there. And you can get things like this. So here's some leaf scans. So we put them inside of this kind of bounded area because we're going to crop out just the leaf. And then there's a computer software that turns this into a black and white image and then measures the number of pixels that are black. And that's how we calculate the leaf area from this. And we use a known distance. So like we know the scale because we have a ruler here. And then we save those leaves in a coin envelope. So here's, some of the leaf morphology you can imagine, like when you get a woodsia, you chop it, chuck it on the scanner, scan it, move on. Awesome. Erigeron composite is slightly more complicated, right? You got to like really try to peel apart those dissected leaves so that you're getting that full leaf area. And then the worst of the worst or the best of the best, depending on your opinion, is these things with like tiny needle-like leaves, which are just, yeah, you got to dissect everyone and get it on there. And we put on multiple leaves and then we divide by the number of leaves because we don't want to just rely on one measurement from one tiny leaf. Okay, so I want to show some super preliminary results for just a couple of traits about what's happening across these gradients. And again, this is combining the community data from the downslope transects and the trait data that I collected in the field, putting those two together. And then for each transect, making an average trait value. And then that's what I'm going to show you in a second. I don't know if I animated these. Okay, try to just look on the left. So each color shows a different peak, right? So I showed you there's these 5 peaks. The y-axis shows the plant vegetative height, and each one of these points is a belt transect on a given peak. So this point right here represents a belt transect on Campedo Mountain that has an average. If you take all the vegetative heights across all the species are there, and you make an average of them, the average vegetation height down on Campedo on this belt transect is about 12.5 centimeters. So there's some tall plants there. And if we go down here, we're looking at White Mountain Peak. This dot right here, right, way up high, the average plant height is less than 5 centimeters. So that's what this shows on the left. On the right, same plot, just with a different trait, and that trait is specific leaf area. And that, again, that's the ratio of leaf area to leaf mass. So leaves down here are smaller and thicker, smaller area, but thicker, smaller and heavier. And leaves up here with these higher specific leaf area values are larger and thinner. So a couple takeaways from here. One is we got these, this is a good thing, right? That we confirmed that plants get taller or plants get shorter as you go up in elevation. Okay, we like that. That seems like something we should be able to get to pretty easily. The second thing to see, I think, takeaway for me in this left-hand figure is that the relationships, so each one of these lines shows the statistical relationship, like a regression between these two variables. And the dashed ones mean that it's not statistically significant, so not different from 0. When we're thinking about the signal versus noise, I won't do a full stats briefing for you here, but these are not statistically significant relationships. And the full lines, the complete lines, are statistically significant relationships. And one kind of takeaway here, and again, this is just looking at space, so this is only looking at community data for 1 year, is that the relationships are strong if you look across all the peaks between traits and the environment, but that if you look within any one peak, the relationships between traits and the environment, like looking across individual downslope transects, is weaker. And we see the same thing with specific leaf area. Specific leaf area is pretty interesting. I think I might have expected, and I could have tried this out on all of you, but and maybe I should have. I think I would have expected that plants at higher elevations would have smaller, thicker leaves. We found the opposite, that plants at higher elevations have like larger, thinner leaves. And I think there's some cool maybe reasons for that. One might be that plants up here have this like limited growing window, so they're like making these thin leaves quickly and trying to get as much out of them as possible. Another idea is that the plants down here, that we're kind of crossing this water temperature and water gradient. So maybe down here, plants are really limited by water, and so they need these small, thick xerophilous leaves to withstand those dry habitats. And then up here, temperature is more limiting and water is a little less limiting. And so plants are having these larger, thinner leaves to kind of get as much as they can out of those leaves. So just a couple of things we can do with this. I think the next thing we can do with this, which is really exciting to me, is we can look at this over time, right? So we can make each one of these, just for example, we can make a figure like this for every single year that we have on each of these. So we have multiple surveys for each peak, and we can think about how functional trait values are changing over time. And I think one of the hypotheses from that work, or for that analysis, is that we might be expecting that these plant communities are getting taller over time. So maybe plants are able to grow taller because it's warmer on those peaks. But to be determined. And I think another thing I'm excited with this is kind of comparing plant community changes, like compositional changes, taxonomic diversity changes, and thinking about how those explain what's happening there versus these, or in addition to, in complement to these functional trait analyses, and seeing you know, what information is complementary between those two facets of biodiversity, and what information might we learn if we look at either of those. So yeah, I'll be interested to hear your thoughts about this. So I'll just wrap up by saying I've said thanks a bunch during this, and I that's really important to me. But thanks to all the people and all the organizations and institutions that have funded this work. Again, just huge shout out to my advisor, Seema Sheth. She's been so supportive of me, and it's been super beautiful to be here in her lab at NC State, and feel really lucky to be part of GLORIA Great Basin. Bunch of other organizations have supported this work. And so, yeah, huge gratitude to them. This QR code will take you to the GLORIA Great Basin website, which I sometimes help to manage, and has a whole bunch of information about this project. And one of the crucial bits of information about this project, so you can, if you put your phone's camera up to this, it will take you to the GLORIA Great Basin website. So you can do that on the recording, or you could do that now. And on that webpage, you'll find this, which is to say that this work is ongoing, and something that for me is super inspiring about this work is that the goal of it is to truly be long-term, and to truly bear witness to these places on timescales that sort of surpass the timescales of our daily experience, right? So I think I already said, right, that, you know, when people started looking on these summits, or when GLORIA scientists started looking on these summits, I was in 5th grade, and right now I'm here, and I'm like, I'm going to take this, you know, as far as I can. I'm going to take this as far along in my life as possible. And I think all of us who are involved in this are hoping for another 50 or another 100 or even another 200 years of sort of bearing witness to these places and exploring these places deeply. So every year we're out there doing that. Every summer, and I'm involved in sort of getting people together. So this summer we'll be in Death Valley resurveying some summits the last week of June. We'll be in the White Mountains. These stars mean tentative, so yeah, this could change. But we'll be in the White Mountains 3rd week of July, and then the last week of July we'll be in Great Basin National Park in Eastern Nevada. We would love to see you. We would love to, if you're interested in, you know, being part of this work or supporting this work, definitely, you know, send us an email. I got my email on the next page here. So thanks so much for your thoughts and attention. I'm excited to hear your questions, comments, mysteries, and revelations. And I'll just leave you with this little quote from Turtle Island. So thank you so much.
Well, thank you very much, Caleb. And are there questions from the group? You can unmute yourself, or you can type them into the chat. Whatever works.
And I'll maybe just keep sharing my screen in case someone needs me to go back and forth. So yeah.
I have a quick question.
Sure.
This is Alan Bade and Wendy Gollop together. I'm curious if you measured the movement of invasive species upslope, you know, in your surveys. Are you seeing species being pushed out by invasives that are moving upslope as well?
Yeah, in the Alpine, in the Sierra Nevada and Great Basin, we haven't seen a ton of that, especially on summits. I'll say, just from my experience as a park ranger in Tuolumne Meadows in Yosemite National Park, that there are species that are non-native and even invasive that have sort of shown up at 8,600 or 9,000 or even 10,000 feet. Some of the ones that strike, there's a hypericum, I think, that's up there in Tuolumne Meadows. There's a Sprigularia, which is kind of a weird one. In the GLORIA work, we've seen a few instances of seeing kind of scary things on top of summits. So like there's been a couple of observations of Bromus tectorum on summits, like single individuals, which I think is really interesting, and sort of proves the point that like those things can exist there over, we don't know how long can they stay there. But I think that is part of this work, is to be there and just be like, what is here year after year? And so far, yeah, we don't have like a strong signal of invasive, or kind of, yeah, your kind of classic invasive or non-native species in general, right? The idea is that it's just really hard for species that aren't locally adapted or aren't adapted to those high elevation places to be in those high elevation systems. But as things change, my expectation would be that, yeah, more things from lower down and even way lower down, like, thinking about cheatgrass and things like that, can make it to these summits, especially as things get warmer there. So that's just a spitball kind of answer to your question, maybe with a few observations from what we've actually seen in the field. Yeah.
And actually, I would have a follow-up on that. When you do see these invasives or exotics, do you remove them?
So this work happens in collaboration with lots of land managers across Western North America. And when we are working on public land and with those land managers under their permits that come from those organizations, they sort of set the rules. So I know, for example, in Death Valley National Park, which is where one of the Bromus tectorum individuals was discovered, the land management person that was there, the person from the National Park Service that was there, wanted to remove it right away. And I think people who are responsible for public lands, that is their kind of, they're like, we're not dealing with that, like people who deal with that as part of their career. Yeah, maybe there are some of us who as scientists are like, what would happen? Like, would we find this again in 5 years? Like, we don't have the answer to that question because, yeah, a lot of times those things have just gotten yanked right away. So yeah, that's the candid answer to your question. Yeah.
Okay, actually, I'm glad to hear that they were removed.
Yeah.
And then in the chat, Bob Case writes, wind exposure, temperature variations over season, hard micro niche variables, insect pollinators and insect grazer populations. Try to get some Beartooth Plateau data, largest North American alpine. Isn't the largest North American alpine actually the Uncompahgre Plateau in Colorado?
I don't know. That's cool. No, I'll check out the Beartooth Plateau. Yeah, that's super interesting. And yeah, I think there is a cool aspect to be built into this that some people have worked on and maybe not enough has been done, which is sort of thinking about these we have all these cool ideas about islands in ecology, right? We have island biogeography from MacArthur and Wilson. And I think that those topics are very relevant here in the Alpine, where we have these sort of sky islands. So thinking about, the area of those islands, how many species are there? A little bit of stuff has been done there, and we sort of have these nice predictions based on first principles. But I think that's super interesting. Yeah, like what's the biggest? How many species are there? What's the smallest? How many species are there? I mean, in Yosemite, you can do that entire thing just in Yosemite National Park, right? Because you have those massive unglaciated plateaus, huge sky islands like the Parsons Plateau and the Dana Plateau, and then you also have these nunataks. So yeah, there's some people out there I know who are interested in that, and I think more power to them. It's an awesome question. Yeah.
Okay, and then another question from the chat from Kevin. Are there confounding factors besides temperature change that affect alpine plant changes, such as change in wind speed or precipitation?
Yeah, this is a great question, and I think, yeah, I could do a better job here. And I think just in my own thinking of kind of being as holistic, I think we should be as holistic as possible of thinking about like all of those different kind of co-varying abiotic dependencies that are in the Alpine. And certainly all those matter. And I think one of the ones that's, I don't want to say easy, but one of the ones that our mind goes first to, right, is temperature. And I try to show some of those those are very broad kind of climate model answers to the question of like, how much warmer has it gotten? There's some more kind of fine scale analyses out there. But it has definitely gotten warmer on those summits, even just like in the last 20 years, and in the last several decades. I think precipitation matters a lot. Actually, kind of like the whole, I think one of the keys to understanding this place, the Sierra Nevada and Great Basin particularly, is thinking about this interaction between temperature and precipitation, right? Because you could have an increase in temperature and a increase in precipitation, and you get this kind of, like tropical effect, right? Where it really becomes easier, or you could have an increase in temperature and a decrease in precipitation. And that's a completely different trajectory. And there are some awesome people out there. Big shout out to Megan Oldfather, just published a beautiful paper from with colleagues from Niwot Ridge in Colorado, where they think about these divergent community trajectories, where like the communities are going in two different directions. One of the communities is under more snow than the other is kind of the simplest way to put it. So I think those all matter. There's been some recent like global analyses about how wind will change and has changed with climate change. So like the global wind patterns are also changing with climate change. And so, yeah, when you start putting these things all together, you really start to see the complexity of these places. And yeah, there's the complexity of each variable interacting with each other, but there's also all of this complexity within each variable. And I think, a big point to make in this system is that the air temperature, right, the air temperature at 2 meters, which is like where a weather station would measure temperature, is totally different from the temperature that an alpine plant, and especially like an alpine cushion plant, experiences. And so, yeah, we should be as specific as we can. Although to say too that at a certain, if we go too deep down those rabbit holes, it keeps us from doing anything sometimes. So yeah, trying to strike that balance. That's a great question.
And then a couple of things occurred to me. One that we had talked briefly about before the talk really started, and that was about annuals, alpine annuals. And that I noticed you really didn't go into those sort of at all as far as even your classifications of cushions, subshrubs, forbs, etc. And again, given that the Sierra Nevada and sort of some of these more desertic alpine areas are known for having more annuals than any other alpine areas. I was wondering why that wasn't or isn't a focus, and especially since you mentioned Bromus tectorum being up there, another annual. It would seem that annuals would be something to be watching in a period of climate change and global warming.
Yeah, no, I think that all the questions about annuals are super interesting. And I think my interest in the questions about annuals are from kind of evolutionary biology perspective, right? So evolutionary biologists like really like to know the fitness, the lifetime fitness of an individual, right? So we can like do lots of cool stuff with that. I have thought a little bit about how to do that with androcracy, septentrionalis, like we were saying before this. For these like big kind of broad scale community analysis, like if you were to take the entire GLORIA Great Basin species list, like just everything we've seen, and you look how many annuals are on the list, there's like 3 or 4. And it's hard to do. So like they're out there, but like we maybe haven't captured them all yet. And I think that's that's part of this work. But yeah, it'd be hard to do. It would be hard to split them out because there are so few that we've seen in these surveys on these summits. But I think there's all these other interesting things that you could do with alpine annuals that I hope people will do, especially in our area. Yeah, and actually Androcracy septentrionalis was used in a really beautiful study in this place in Colorado at Rocky Mountain Biological Lab, where they've been warming these meadows. John Hart and colleagues have been warming these meadows with like a consistent amount of infrared radiation. They've been warming them for like 40 years or something. So this person was able to like do all this cool demography with androcracy and think about kind of how annuals might be impacted by climate change. So yeah, there's too many cool things, too many beautiful things.
Yes. Okay. There's another comment in the chat, and it's from Aaron saying, "I think Bob may have meant to emphasize insect-plant interactions, pollination, and herbivory issues." And then parenthetically, I know Michael and Ellen Euler have found high elevation endemic orthopedic or orthopteran high in the Sierra.
Yeah, I say let's go, let's do it. Yeah, I mean, there's a couple of things out there I can think of. There was a thing, I think it was in Science recently, about kind of how communities could be impacted by herbivory in the Alpine, and that was done in the Alps. I could probably send that, and you could send it out on email. But yeah, I think it's probably super important and super interesting. One just like tiny story I'll tell about high elevation arthropods, or about insects specifically in this region. A friend of mine works on gray crown rosy finches in Erica Zavaletta's lab, and he's interested in diet of gray crown rosy finches up in those high elevation places, and they eat insects off the surface of snow that's stuck in summer way up there. And he and his colleagues scraped a bunch of insects off of those snowbanks, and then they weren't sure how to identify all the insects quickly, and so they posted them all on iNaturalist, and they all got identified like super fast by all these experts. iNaturalist is amazing, of course, and iNaturalist I think is where I go, and where other people go to like find weird stuff, right? If you're interested in like weird, yeah, if you want to just kick back after this meeting and probably find some species of insect unknown to science, just go on iNaturalist, it's all there. But they found that the insects that the gray crown rosy finches were eating at high elevation, most of those insects were actually blown up, like blown up from the valley, and they were these like fat alfalfa insects you find in alfalfa fields, and they'd blown up there and then stuck to the surface of the snow, and then the rosy finches are eating them. So there's also like, yeah, there's also these, when you start to think about animals that move around, yeah, the connections even grow more than that. So no, I think it's cool. Another kind of story about insects that I'm interested in, and I don't know how to get this out there, but like again, going back to Holsia algida, to the Alpine gold, right? It has like almost like Drosera like hairs. Like it is like so sticky with trichomes. And when you look at that, if you look at herbarium specimens, or even if you look at wild plants, those trichomes on Holsia are covered in flies and gnats and midges. I don't know. I mean, I don't know anything about that. I don't know if anyone knows anything about that. Like, is Holsia some like half parasitic? I mean, I don't know. Maybe there's probably some like real entomologists out there that are laughing at me right now. But yeah, I think that it's people like Michael and Ellen, and people who are out there on GLORIA surveys, and people like you all, who are posting things on iNaturalist and taking pictures, and they're the naturalists who are out there crawling around finding things, because there's still so much to find, and there's still so many observations like that without a story, and they should have those stories. So yeah, I appreciate that question a lot.
Yeah, and that I remember reading, well, this is quite a while ago now, that that the viscid nature of proboscideas were documented to actually be dissolving the insects that got stuck in them. And so it got classified as an insectivorous plant,
Yeah.
or sort of, entry level, or perhaps in that, or developing more in that area.
Mhm.
One other question from your slides, and then I see Bob has another comment here, is that when you were talking about the different plant types, and the slide said that there was an indication that cushion plants were decreasing. Could you speak a little bit about that?
Yeah, sure. Yeah. So I did some analyses with abundance data, and the abundance data that we have for this data type that I showed in that kind of first section are these 5 categories. And that's part of the international protocol, right? So we asked people to say, like, is this plant in this plot common, dominant, scattered, rare, or very rare? We ask people to put them in there, and there's some kind of nice definitions. And I made this kind of I made a statistical model that can kind of deal with those categories. And the output of that was this kind of weak decrease in cushion plants. So yeah, just to say that abundance definitely matters. And, when you see a result like that of like, oh, there's, not much change. If you buy that, not much change in taxonomic diversity or in species richness over time, we start thinking about like, what are the patterns of turnover? What are the patterns of abundance? Like, for me, when I see that, I'm like, oh, the action is in some other part of community composition. So I think abundance is something that I'd love to look more at. And part of the GLORIA protocol are these more like quantitative ways of thinking at abundance at a little bit smaller scale. And I think those could be super powerful at like actually being able to put a number on those kinds of declines, and I think also, or possible declines, and also, kind of checking our work using multiple data scales is what we would like to do. So yeah, just putting that out as like tentative, that is what came out in this model. More work to be done. We've seen some interesting things in the field, like we've seen big areas of cushion mortality in the field when we've been out there. And that's a hard, if you see a few dead cushion plants here or there, you kind of think like, oh, was that like that last time? Like, oh, I'm not sure I remember. Like, is that how it's supposed to be? Like, so I think, it takes us a while to like really start to have the data ready to answer those questions in like a quantitative way, rather than just like, these are some things we've seen. So yeah, that's the best I could say about that. I see Michael with his hand up.
Yeah, well, and that, but do you have
Yeah.
any thoughts about why that might be? Why the cushion plants may be having more of a problem than other growth forms?
No, I don't think I've thought about
Okay.
it enough
All
yet.
right, well, that's certainly
Um,
fair and
yeah, yeah.
honest.
But I think it's a functional group that really matters up there, right? And we
It
think about
does.
facilitation and all those things. And so that's a group of plants that I'm sort of constantly looking at and thinking about what other species are inside of those cushions, and is there mortality on cushions? Are there insect interactions on cushions? And just, I guess maybe just a plug for all the people here, right? So next time you go walking in the Alpine, like check out look at the cushions closely. I think they're part of kind of the key to those places and them being what they are. So
Yeah,
I think
well,
Michael Euler has a question.
yeah, go ahead, Michael.
Yeah. Hey, Kaleb, I just want to say personally, thank you so much for being here and doing this. Your research is like the most interesting thing going on in my life. And I love the Alpine and I want to spend more time there with you. And now I have more things to look for. Every time I go there, I want to be looking and recording. So yeah, once again, just thanks so much. I look forward to seeing you this summer.
Yep, me too. Yeah, can't wait. We'll all be there. Thanks, Michael.
okay, and then the other thing that Bob typed in was that the Beartooth region is above 10,000 feet and Uncompahgre is above 9,500 feet. And yes, that's correct. I think that Uncompahgre is considered to be the larger area, or the largest area of alpine vegetation, contiguous alpine vegetation outside or in the conterminous contiguous US. And then I see there are two more messages. Ellen typed in, Ellen Euler, so glad that you're doing this incredibly interesting work and are standing by to support you as able.
Thanks,
And
Alan.
Kevin wrote, good work, especially at high altitude oxygen levels for us humans. Do you know if anyone is doing artificial climate change studies in laboratories?
Yeah, people are definitely doing that. Yeah, sort of like trying to put there's cool stuff everywhere, but there's a cool thing I'm thinking of from Europe. There's a lot of research in this area, like alpine community ecology, where people take intact communities. And so what that looks like is like a turf transplant, which if you look at the slide, is not possible in most places where we work. I mean, there are some places where maybe you could dig out a turf, but it would be kind of a slice of like it's kind of like one microhabitat of the Alpine if you try to do that in our place. So it's harder to draw big conclusions, but there's a lot of research with alpine grasslands, especially in Europe, where you could cut out a turf, and then you could take that turf to a growth chamber or like to a greenhouse, and you can expose that turf to all kinds of crazy stuff, and then or the climate change predictions, and then measure how those communities change over time. People have done really interesting work with that. Just even one thing where someone took some turfs, move them down to simulate a heat wave. I didn't talk about like these extreme changes, kind of changes in variability. And I think, one of the for sure things we know that climate change is causing is an increased incidence of extreme weather events. And we've all seen that. You can see that in California really easily. And there's this idea that can cause communities to do different things and go in different trajectories. And so someone did this cool thing where they took those turfs, moved them down to simulate a heat wave, kept them there for a couple weeks, moved them back, planted them back in their holes, and then monitor those communities over time for a long time. So there's all kinds of cool things that you can do like that. Yeah, and people are definitely thinking about that, and I'd love to get in on that as I can. As a naturalist, it's hard not to do these observational things, but yeah, those experiments are powerful and are out there.
Okay, well then, are there any other questions or comments? Alan and Wendy typed in, great talk, thanks.
Hi, Caleb. I'll say the same thing. It was wonderful.
Thanks for having me. Yeah, thank you all so much for having me. This is a real pleasure.
It's very impressive, Caleb. Very impressive.
Yes, so thank you very much, and hopefully you'll be out here again and can give us more updates sometime in the future.
Sounds good. I really want to see the Alpine garden that y'all have there. So yeah, thanks so much for what you all do.
Sure. Well, again, thank you very much for the talk today, and for everyone that's still here. Remember that next week in person, we have Michael Euler speaking on another one of his Sierran adventures. So please join us in person. We will have masks available for anyone that forgets one or feels they need one, because especially since we're having more COVID show up in our region. So with that, thank you very much. Again, Caleb, and thank you all for being here, and hopefully see you next week. Bye now.
Okay, I'll stop the recording.
Thanks.